So I've showed you already data from our laboratory related to use of the vesicular monoamine neurotransmitter transporter for imaging presynaptic dopamine nerve terminals. I'd now like to share with you a little of the basic science and method development underlying those data. So again, here's our high-resolution nerve terminal. And again, we're going to focus on the imaging of this transporter as our index of how many of these dopamine nerve terminals are present. In the next slide, I'm going to show you, though, a comparison between use of AADC and the DAT for imaging early Parkinson disease from Tedroff and his colleagues in the late 1990s. And what you see here is a normal control individual, and what's depicted in color is the ratio between the fluorodopa and the DAT uptake binding. And you can see that throughout the striatal complex, there's kind of a nice orangey color. Maybe there's a little more fluorodopa signal than DAT in some parts of the putamen. Contrast this, though, with early, mild Parkinson disease. You can see that there's relative preservation of the fluorodopa signal, so now this putamen becomes relatively yellowish compared to the DAT. And this indicates to us that there's counter-regulatory change in the expression or activity of the DAT protein versus the AADC. And it's for this reason that our laboratory went forward to look for an additional marker or markers of these nerve terminals that might not show this compensatory regulatory effect. Now, talking about vesicular neurotransmitter transporters in general, all neurons that use small, low molecular weight neurotransmitter substances have transporters on the synaptic vesicles that move the transporter from the cell cytoplasm into the synaptic vesicle. And this is true, of course, for glutamate and other excitatory related transmitters, for GABA, for acetylcholine, and we'll talk about that later this morning, and for the monoamines. We have tracers for two of these. One of these I'll show you a bit later. But we're going to focus now on tetrabenazine and its analogs for tracing of this transporter. We do not have existing imaging methods for these vesicular transporters, and we would really like to have some. So these vesicular transporters have been characterized now on a molecular basis. They're 12 transmembrane spanning proteins with a large intravesicular luminal extension, and with at least 3 domains that are associated with recognition of and the transport of monoamines. And this is kind of how we think these transporters work. So here's a monoamine, in this case histamine. It binds to the transporter, and then the transporter flips and deposits the monoamine on the other side of the membrane, and then this is recycled. The recycling, though, of the transporter back to the cytoplasmic side requires a hydrogen ion. So this is a pH-dependent cycling, and it's the hydrogen ion concentration across the vesicular membrane that drives the active transport of monoamines into the cytoplasm. And then you can see that both tetrabenazine and reserpine block the ligand recognition site and prevent the transport. I guess the other thing here that you're supposed to pay attention to is that the action of reserpine requires that the transporter be inserted in a membrane and have this hydrogen ion activity across the membrane, whereas tetrabenazine binding is reversible and does not require that the protein be inserted in a functional vesicle. Now, in our laboratory, we were very interested in addressing whether damage, and particularly death of striatal neurons, was associated with a quantitative estimate on the basis of VMAT2 binding. And a fellow in my laboratory did hydroxydopamine lesions of the substantia nigra pars compacta, actually of the nigrostriatal pathway. And this was quantified by tyrosine hydroxylase immunohistochemistry. So here's the control side of one of these rats, and here is the lesion side in an animal that had a very severe lesion. And in an animal like this, where we have virtually complete loss of the dopamine neurons, we see a virtually complete loss of specific binding of tetrabenazine to the vesicles. So here's the intact striatum, and here's the denervated side. Now, one of the well-recognized but not advertised aspects of hydroxydopamine lesions in experimental animals is they're somewhat difficult to do in a reproducible fashion. The structure that one is trying to inject the toxin into is very tiny, and a very small error in positioning of the injection catheter can result in entirely missing the nigrostriatal projection. So what we see here are across a number of animals that have a binding asymmetry from practically none, so these were missed, to some that have a very severe asymmetry. There's a linear relationship between the tetrabenazine binding in the striatum and counting the number of dopamine neurons remaining in the substantia nigra. So it looks to us like this is a very reliable measure of death and damage to these dopamine neurons when the damage is permanent. On the other side, we want to know, do non-lethal interventions that change the functioning of the dopamine synapse result in a regulatory change in VMAT2 sites? And so what you see here are control animals, animals treated for several weeks with high doses of apomorphine, a dopamine receptor agonist, animals treated for several weeks with high doses of haloperidol, a dopamine receptor blocker, and animals treated with a drug mazindol, which is a blocker of the DAT transporter site. So what you see represented here in the top row is VMAT2 binding. So the vesicular transporter is not changed by exposure to any of these drugs for several weeks. Whereas dopamine D2 receptors are known to up and down regulate. So if we agonist stimulate the receptors, they down-regulate their numbers. If we antagonist treat, they upregulate to try to compensate. If we look at the DAT, it too it shows counter-regulatory change, and particularly when you block it, it upregulates to try to overcome the blockade. So we think that the DAT itself shows this regulatory change now over weeks to months, whereas VMAT2 does not. And so we favor then the use of VMAT2 as an approach that's insensitive to changes in the activity in the synapse, and instead is very remarkably sensitive to whether the projections are damaged or intact. Now, one of the very controversial aspects about Parkinson's disease, at least several years ago, was whether treatment of Parkinson's disease actually accelerated damage to the remaining substantia nigra neurons. And we were able to address this with the use of VMAT2, where we intentionally now made some incomplete lesions of the nigrostriatal pathway on one side of the brain, and then we characterized the lesion by use of apomorphine injection, which releases dopamine from presynaptic dopamine terminals. And when one does this in a rat, the response of the lesioned rat is to turn in circles. And the more severe the dopamine asymmetry, the faster the circling. So what you see here are individual rats that were lesioned, some of them not at all, so we didn't really get these guys on target, to some that were more severely affected. And you can see that we could predict how affected they were by how quickly they rotated when administered apomorphine. So what we then did was to assign pairwise definitions of these animals based on their rotation to receive a month's treatment with either levodopa, the direct converted to dopamine agent that some had speculated might be toxic to nerve cells, versus a dopamine agonist, which some had argued might actually protect dopamine nerve cells. And here now, from pairs of these animals, is the result on a VMAT binding, and now what we're showing here is the VMAT asymmetry, or the VMAT difference, between animals that were treated one way versus the other. And what you can see is that there's no significant loss of VMAT binding sites from animals that were differentially treated. So at least in this model, we conclude, the same which has now been concluded, I think, from clinical trials, that there is no evidence for neurotoxicity when using levodopa to relieve incomplete lesions, including those in mild early Parkinson disease. Now, there are other interesting aspects of vesicular neurotransmitter transporters that support our notion that these are not aggressively regulated sites. This is an experiment that comes from LNaudon and his colleagues and Dr. Costentin in France, where they administered a single very high dose of Reserpinean to experimental animals, to rats. Reserpinean binds to the transporter and inactivates it permanently. So it alkylates the protein and takes it completely out of action. And what they showed was that a single high dose of Reserpinean depleted dopamine levels and blocked tetrabenazine binding in the striatum very, very, very severely. This hashed bar shows measurement of the dopamine reuptake transporter, or the DAT, on the basis of ligand GBR12783. What you can see, though, is that over a period of time, and in this case up to 60 days after the lesion, the dopamine levels returned a little bit. In fact, they returned to about 50% of normal. The vesicular transporter measure stayed severely depressed. It improved by a factor of about 4-fold from day 3 to day 60, but by no means did it return to the pre-Reserpinean levels. So this suggests that the vesicular transporter does not increase in its rate of synthesis, despite that it's been completely inactivated by this drug. So how is this possible? Because we know that synaptic neurotransmission is a very heavily regulated biochemical process. There must be some explanation for this that allows us to make sense of regulation from the standpoint of function versus what we now believe we know about the protein and its level. And the explanation, we think, comes from studies pioneered by Paul Greengard and his colleagues, related to the functionality of other vesicular proteins, and particularly this one, synapsin-1, which is a protein located on synaptic vesicles, and which regulates the binding of the vesicle to actin filaments in the presynaptic terminal, and thus putting those vesicles that are bound to actin into what's known as the reserve pool of synaptic vesicles. So the idea here is we have a synapse. There are many vesicles in the presynaptic terminal that are inactive. They're bound to actin filaments via synapsin, and then there are other synaptic vesicles that are free to dock with the presynaptic membrane and to release neurotransmitters. So these are in the active or cycling pool. And that the way synaptic regulation or synaptic transmission is regulated at the level of vesicles is not to change necessarily the number of vesicles or vesicle transporters, but to change the number of vesicles that are in the active cycling versus the reserve pool. So if you want to increase the release of neurotransmitter, you dephosphorylate synapsin, and you increase the phosphorylation of synapsin, releasing more of these vesicles into the actively cycling pool. And conversely, if you decrease synapsin phosphorylation, then you put more of these into the bound pool and reduce the number that can be released. So the VMAT2 binding site, just to summarize, is shared across all monoamine neurons. It's not specific to dopamine, but as I said already, in the striatum you can assume that virtually all of the signal is indeed dopamine in origin. Losses in VMAT2 sites are linearly and directly proportional to irreversible damage to these dopamine neurons, but the overall density of VMAT2 binding sites is not readily altered by changes in the activity in an intact synapse, whether that's by compensatory change in response to drugs, or whether it's a function in remaining nerve terminals after a partial lesion. So, how did we target then imaging of these terminals? Well, we decided that we would use tetrabenazine derivatives to target the VMAT2. This is an initial study done in an experimental rat after injection of methoxytetrabenazine, a slight derivative of tetrabenazine. And this shows in a sagittal view of the rat brain intense accumulation of radioactivity in the striatum. And if you take the adjacent section to this and wash it in saline to remove unbound activity, there's very intense retention of activity in the striatum. You can even see the substantia nigra here, and the locus ceruleus as well in the pons. With regards to the nature of radioactivity after injecting systemically, virtually all of the activity in the brain is the authentic radiotracer, so there's no evidence of labeled metabolites, whereas in the blood and in the liver, the drug is metabolized to polar substances. We showed in human volunteers that the binding of tetrabenazines are stereospecific, so the plus isomer is pharmacologically active and shows nice striatal signal, whereas the minus enantiomer shows no specific binding, and that's because it's pharmacologically inactive. What this allows us to show, though, is that some regions of the brain, and particularly the occipital cortex, even with the plus isomer, may be a very good location to find nonspecific binding for a point of reference, and that's actually what we use the occipital cortex for. I've shown you this as well, and this, but here's the quantification now of binding in 30 patients with Parkinson's versus similar age normal controls. And these values on top show the binding relative to arterial blood. And you can see that the occipital cortex, which we've used for a point of reference, actually goes down a little bit, but not significantly in Parkinson's disease, where there are highly significant reductions throughout the striatal complex in Parkinson's. And I've shown you these just previous, but the new information here is that there is actually a correlation between the severity of the lesion in Parkinson patients with some aspects of their clinical status. Unfortunately, commonly used measures of disease severity, such as the Hoehn-Yahr scale or the UPDRS motor rating subscale, do not correlate highly with remaining VMAT2 binding in the striatum. However, disease duration shows a strong relationship to VMAT2 binding in Parkinson's disease, as do some measures of overall daily activity abilities. And so here's an illustration of one of those. So here's the duration of symptoms of Parkinson's disease in years, and here is the degree of reduction of binding, where this is a high reduction and this is very little. So this is highly significant. This shows right hemisphere, left hemisphere asymmetries in binding are reflective of the side of the body and how asymmetrical the movement disturbance is. We also can show that over time, Parkinson's is an active process, and that if we scan patients at entry into our research, and then again 2 years later, we see a progressive loss of binding in virtually all Parkinson patients. And interestingly, the amount of that change doesn't have to do so much with the clinical course, but it's entirely dependent on how much binding was initially present. So the patients that have the mildest lesion, despite that it doesn't correlate terribly well with clinical measures of severity, declined most, and those that had the more advanced lesion at entry declined less. One final aspect of things we've looked at in our laboratory with these transporters are to look for potential neurotoxic effects of drugs, and particularly drugs of abuse. We know in experimental animals, if we give a high dose of methamphetamine, this is associated with a lasting reduction in dopamine chemical signaling, and it's manifest by reductions in binding of VMAT2. We looked for evidence of the same sort of change in humans who had been abusing methamphetamine, and for this we went to San Francisco, where there was a population of psychostimulant abusers who were well known. And what we found is that in these psychostimulant drug abusers, but who had been abstinent in most cases for more than 2 years and some more than 10 years, is that they had a statistically significant loss of VMAT2 binding throughout their striatum, but kind of the opposite of Parkinson's disease. The more strong, the largest reduction was seen in the caudate nucleus rather than in the putamen. But this is evidence that exposure to these psychostimulant drugs on a repetitive basis does indeed produce damage to the brain. Just as the last slide, these are my colleagues who participated in the development of the work that I've shown you. This is Mike Kilborn, our radiochemist, who conceived of the tetrabenazine analogs. Bob Kepi, our medical physicist, who works on compartmental modeling with me. And these are 3 fellows who actually measured all the data I've shown you. This is Thierry Vanderboort from Belgium, Satoshi Minashima from Japan, and Nicholas Bonin from the Netherlands.